antibiotics, and also for other small molecules, as shown by the first presentation seen today. Streptomyces has antibiotic resistance mechanisms that it does not use. For example, it has many genes that bear striking similarities to the 3 different types of tetracycline resistance. However, Streptomyces silicolor is actually sensitive to tetracycline. There's a mistake in our slide. Therefore, these genes must operate on other antibiotics or metabolites within the cell. So having a better understanding of these mechanisms could help us better exploit these Streptomyces silicolor. And this is why Dr. Nodwell received many of his research grants. So if you'll remember from last time, we used the TET-A, TET-R system of gene repression to serve as a model for our experiment. Here we see that the TET-R protein in red is dimerized and binding to the operating sequences of both TET-R and TET-A, repressing their transcription. Upon the introduction of tetracycline to the cell, tetracycline comes in and binds to the TET-R dimer, causing a conformational change which will release the TET-R dimer from the operator sequence, thereby relieving repression of these genes. The TET-A gene codes for a tetracycline efflux pump, which will then be translated and localized to the membrane, where it will cause the export of tetracycline from the cell, thereby conferring resistance. So the system that we're studying today is SCO4870, SCO4871 system, as TET-R and TET-A has been extensively studied. SCO4871 encodes a TET-R-like gene which contains a helix-turn-helix motif that is very similar to TET-R. SCO4870 is a putative monooxygenase that looks to be very similar to the TET-X protein. We also identified 2 putative operator sequences that are divergently oriented in between the 2 genes. Also, I forgot to mention in the last slide that the TET-R, TET-A operator sequences are palindromic, and from the orientation and palindromic characteristics, we identified 2 possible operator sequences for this system. So the objective of the experiment was to devise a 2-step biosensor system that would allow us to study all TET-R-like proteins using our SCO4871, SCO4870 gene system as a model.